Alrighty. So I was just going to give a brief introduction into my career and background, but I think Heidi covered most of the points there. So I'm just going to skip right to this part, except initially, before a few people joined, I just mentioned about how my interaction with Swiss started. So I actually went looking for Swiss when I moved to Canada after I did my PhD in the US, and I wanted to get involved with an organization that was working for improving increasing involvement of women in STEM fields. That's how I found Swiss. I started volunteering with the membership committee in early 2017, and then I was elected to the board 2018, 2019 as the director of IWIS. I'm looking to hopefully now ramp up my volunteering activities now that I've settled into this not so new role anymore. I should have more time to do that. So hoping that I'll get to see more of you more often. Moving on. So I was informed that the audience is usually a mixed bag of different backgrounds. So I thought I'll just cover some basics first. So what is genomics really? And to answer that, let's just first address the basic concept of a genome. So an organism's complete set of DNA is called its genome. To put this on scale, we're talking about approximately 3.2 billion DNA base pairs of the four letters that make up the DNA code, which is A, T, G, and C. Now, this genome contains about 20,000, 22,000 genes that makes up for around 1 to 5% of the genome. And these genes carry instructions for making proteins that our bodies are built of. The rest of the DNA between the genes is the non-coding DNA because it does not really code for any proteins. It used to be initially called junk DNA as it wasn't thought to be important. But we know now that this DNA is important for regulating the genes and the genome as well. So it can help switch genes on and off at the right time. So there's still a lot more to learn about what it all does. And that brings us to genomics. So genomics is essentially the study of the entire genome and how it works, but also has broader meaning now to include the way the genome is interpreted and the different technologies that have really developed because of it. But how do you study this whole genome? So you basically need something first that allows you to read the DNA code. Sequencing is the technique that does just that. It basically finds the exact order of the four letters of DNA one by one. And sequencing the entire human genome then means finding that exact unique order of those 3 billion letters of DNA. The biggest milestone project that made all the recent genomics discoveries possible was the Human Genome Project, which was an international collaborative research program whose primary goal was to completely sequence the human genome and map all the genes on the genome. The project launched in 1990, and I think the full sequence was completed and published in April of 2003. So this endeavor of publishing that first human genome really took 13 years and cost about 2.7 billion US dollars. Today, sequencing a human genome takes about 2 to 4 days and costs around 600 USDs. So we've come a far way. But then, why really sequence a genome? So one of the biggest envisioned applications of the Human Genome Project was its impact on healthcare. No two individuals' genomes are identical. Change in the DNA sequence is called a variant. At times, these variants have no negative impact, but are the reason we all look different and unique. Sometimes genetic variants can lead to detrimental effects, resulting in low or multifunctioning proteins, which can cause disease. Uh, Geneticists believe that close to about 80% of human disorders have some genetic etiology. So, learning more about genomes can really help us identify the cause of these genetic disorders. Now, some genetic disorders can be caused by as little as just one single variant change. It's kind of like one tiny spelling mistake in someone's DNA. Now, looking at the genome of a person that's affected by such a genetic disease can then help find which DNA changes might actually cause the problem. Now, DNA variants can either be inherited from one generation to the next, as is the case with Mendelian disorders or rare genetic disorders, such as the kind of that I work with, and even some hereditary cancers, or these DNA variants can then be acquired over lifetime, as is the case with certain somatic cancers. So, for some patients, knowing more about their genome may mean a particular treatment can actually be recommended. Now, some genetic variants can make you slow or non responders to certain types of drugs. Now, that is the study of pharmacogenomics, and it's enabled by studying genomics and drug responses across large population cohorts. Also important at the population level is the study of common conditions, which have a basis in genetics, where carrying certain genetic variants or mutations can increase your risk for disease. So, when a genome sequences of those patients with common conditions like, let's say, asthma or diabetes are compared, it's possible to then start seeing certain patterns. Now, these patterns, when studied with health information of those people, can allow us to link those patterns with the risk levels of disease, or even understanding how severe it may be. This can then has the power to inform healthcare as well as health policy. Now, a few years ago, all the technology of genomic sequencing and the applications were kind of restricted to the research environment. However, now genomics is rapidly transitioning into clinical practice, and implementation in healthcare system has been supported by substantial government investment that is totaling over like more than US 4 billion US dollars in at least 14 countries right now. Now, these include massive national level cohorts from countries like US, UK, Australia, and these countries have invested in population based sequencing projects where they return the results to the participants. But there are other countries where national initiatives, for example, in Switzerland or Netherlands or Brazil, and even Finland, are primarily focused on the development of infrastructure, such as common standards and data sharing policies and platforms. It's anticipated that over the next 5 years, genomic data from over 60 million patients will be generated within healthcare. Now, as you can see, there's no such effort happening in Canada right now. So it's seriously lagging behind in this global effort. When you look at the diversity of the approaches that are used by these countries and the progress that they have made towards meeting these challenges of integrating genomics into mainstream healthcare practices at a national level, a common trend emerges in that all these countries initially really heavily invested in developing and testing the necessary infrastructure capacity for sequencing rare disease cohorts, and then kind of scaled that effort into population wide sequencing that was not just limited to rare diseases. Um, so, what are rare diseases and why are they worth studying? A rare disease is basically defined as a disease that affects less than 1 in 2,000 people of the general population. There are between 5,000 to 8,000 known rare diseases right now, and that number constantly keeps increasing every year. In Canada, it's estimated that 1 in 12 Canadians will be affected by a rare disease at some point in their lifetime. That really equals to close to 3 million people. Two-thirds of these children with rare diseases, two-thirds of the people with rare diseases are actually children, and 30% of these children with a rare disease will not live to see their fifth birthday. So even though individually they're rare, when you combine them together, that equals to a really high healthcare burden. About 80% of rare diseases are genetic in origin, but only 50% of them have a family history. So it's tackling a new disorder each time almost, because you don't have a basis to understand what's going on. But because each disease affects only a small number of individuals, understanding and expertise is limited and kind of fragmented across the country. More devastating is the fact that only 5% of rare diseases have an approved treatment worldwide. For Canada, this number is even more sobering. Right now, only 60% of treatments for rare disorders make it into Canada, and most of them get approved up to 6 years later than in the US or Europe. So, overall, people with rare disorders in Canada are really missing out on treatments that could potentially save or significantly improve their lives. And that really needs to change. Steps are being taken in the right direction right now. Genome Canada is sort of laying the foundation to bring this precision health or precision medicine in normal healthcare or clinics across Canada. And their strategy is currently focused on clinical diagnosis of rare disorders, similar to what other countries have done previously. Now, this work is obviously going to be supported by a very collaborative and internationally recognized group of clinicians and researchers. The focus on rare diseases is will hopefully support the development of the necessary infrastructure, the processes, the policies, and the partnerships that will be required for a more broader national initiative. This clinical implementation for rare disease precision health will largely draw from research efforts that have already been successful in this space across a variety of research projects at institutes all across Canada, with Sick Kids in Toronto and BC Children's Hospital Research Institute in Vancouver really leading the way. Now, since the last 2 years, I have personally worked on rare disease research at BC Children's Hospital. These have been collaborative projects between clinicians and scientists, including bioinformaticians as well as basic science scientists. Now, my role has primarily focused on genome analysis of individual families with the goal of identifying the genetic cause of disease. But a lot of the interactions really expand in the day to day beyond that point. And I'll just show you how. So this is a snapshot of the genome analysis process on the research side. Now, it typically begins with a patient suspected of a rare genetic disorder where traditional diagnostic methods have really failed at providing an answer to the family, or a molecular diagnosis has really been evasive. So the doctor will then refer the family to a suitable research program. After consent, enrollment into the study, and sample collection, next generation sequencing is performed, allowing that's then followed by genetic data analysis and hopefully results in the identification of a causative variant and a diagnosis for the family. As a genome analyst or a bioinformatician, my involvement really begins once we have genetic data to work with. I analyze that and I'm involved in the process all the way up to generation of a genetic report. Now, genome analysis involves several steps that need to be customized according to the family's needs, but they sort of largely revolve around concepts of big data management, computing, problem solving, and a lot of collaboration. One second. Okay. So before we get into the details of the bioinformatics workflow, I just sort of want to touch base on the sequencing technology itself because that's sort of crucial to everything. So genetic sequencing is typically outsourced to external service providers. And at that point, there's a choice between the kind of genetic testing that you want to perform or for the identification of genetic variants or mutations. Either you can sequence the entire genome, including both the genic or the coding regions as well as the non-coding regions. Now, this is called whole genome sequencing. Or you can only sequence the coding regions, that's the DNA that actually codes for instructions to make proteins. These regions are called exons, and they cover about 1% of the genome. And the process of sequencing just those coding regions is called whole exome sequencing. Now, the method of choice really depends on the clinical presentation, previous testing, and the diagnostic setting as well. So, are you doing this in a clinical or a research setting? But it's also largely influenced by the benefits and the limitations of both the technologies themselves. So, whole genome sequencing is generally considered to be a far more powerful tool than whole exome sequencing because it can really be used as a single test to capture nearly all known genetic variations, right? So, including your single nucleotide changes, your small insertions or deletions, or changes that span a much larger region like copy number variants or structural variants. Now, compared with whole genome sequencing, however, whole exome sequencing has much fewer storage requirements and the data analysis is easier to perform and historically has also had a very lower per sample cost. And that has kind of traditionally been the method of choice. However, things are now changing and genomes are getting cheaper by the day. So it's really becoming more and more the method of choice increasingly now. So once you have the sequencing data, the next steps sort of include processing and analyzing that data. And no, bioinformatics is not just pressing a few buttons and letting the computer do the job. It takes a lot more than that. In fact, an average computer will sort of break down while you're trying to do this. So you really have to interact with complex, high performance compute systems, or what clinicians like to call supercomputers. So that requires a lot of training about not just how to do the analysis itself, but also being mindful about the data and the system security and good practices that come along with it, because you're working on a space that is shared by a lot of users and also has sensitive patient data. So you have to make sure that you're maintaining the security and the integrity of that data at all times. So after you do the processing comes the trickier part of data analysis itself. Now, a few slides ago, I mentioned that bioinformatics of genome analysis is largely centered on the concept of computing and big data management. So what kind of big data are we really talking about? Now we know that most of us would know that any two humans will share about 99.9% of their DNA. Now, 0.1% seems to be a tiny fraction, but that 0.1% is responsible for the diversity that you see in the entire human population. And that 0.1% also harbors variants that can make a difference between health and disease. But remember that we're talking about 0.1% of the entire genome. So 0.1% of 3.2 billion variants is still 3.2 million variants. Assuming we are working with a more manageable exome data, and assuming that it covers about 5% of that entire genome, we are still talking about the scale of about 160,000 variants. Finding that one causative variant from this huge list is not a simple task. So the Wasserman lab that I work with via the Wasserman's research group at BC Children's Hospital has developed a bioinformatics workflow to really tackle this problem. And it's a semi-automated pipeline that sort of looks like this. Don't worry, I know this is too technical and I'm not going to get into the details here. But overall, we annotate and sort of overlay additional information about the variants and the genes that can then help us narrow down the number of variants to a more manageable number. Now, this workflow is just to show you the relative scale of the number of tools that go into doing this. It's not just pressing a few buttons. I'm hoping that this will provide a more conceptual explanation of what we are trying to do with this pipeline. So while the initial processing really focuses on identification of all variants, so all the letters that differ from your reference sequence, we then leverage additional information to bring down the number of variants for interpretation. So what kind of information does this pipeline really leverage then? Out of all the different tools that are available to us, I think the most informative is doing a trio analysis, meaning ideally we will also have the genetic sequence of both parents as well. And we will try to leverage inheritance patterns to kind of get rid of variants that are common between, let's say, the unaffected parents and the affected child, as they likely do not cause disease. So this cuts down a lot. And that's why a lot of researchers and clinicians will always push for a trio sequencing, even though it turns out to be more expensive because you're paying 3 times the cost as compared to just the child or the patient. Another important one is sort of checking the frequency of that variant or the mutation in the general population. So databases like Nomad have sequencing information from over 125,000 exomes and 15,000 genomes from healthy individuals. So for a disorder that is rare, you want the causative variant to be rare as well. So we typically set our frequency cutoffs around 1% to only include those variants that are present in Nomad at less than 1% frequency. Other variant level annotations include in silico tools to assess the impact of the variant. Is it really predicted to be harmless for protein and gene function, or is it predicted to have a negative impact on gene function? So, you're answering questions like, does the variant lie in an evolutionary conserved location? Has it already been reported in association with a disease phenotype in other clinical databases like ClinVar? Other useful information that really helps is, gene level annotation. So that include information about gene expression and function and the medical phenotypes that are associated with that gene. So for a disorder with a very neurological phenotype, a variant in a gene that's only expressed in the testes is probably not going to be the answer. So all this interaction with all these different databases and softwares and tools happens on a compute system. And most of it really involves me staring at a screen that kind of looks like the screen on the left, the black box on the left. But the end result is a workable Excel file with about 400 variants for exome analysis, obviously more if it's a whole genome analysis. And this information is sort of broken down into meaningful sections. So sometimes I only have to manually review and curate, let's say about 20 to 30 variants before I can find a diagnosis. But sometimes it's a lot more than that. For a whole genome, I've actually manually reviewed and curated, let's say, more than 100 variants for some of the more complicated cases. But it's fruitful and gratifying work in the end. Now, this pipeline has been extremely successful at diagnosing a variety of rare genetic disorders. I have personally used it for 3 different research programs at BC Children's Hospital. Now, depending on the research project and the type of disease presentations it is focused on, the diagnostic rate has varied from 35% to 60%. This is because some disorders are just easier to detect because they have variants in the coding region that are easier to pick up. Especially for neurological disorders, the diagnostic rate is on the higher end, close to 60 to 65% sometimes. And this diagnostic rate is pretty good and very comparable to other published studies around the world. So we are very happy with this pipeline that we built for sure. I'm now going to provide one example of how this genome analysis process has not only provided just a genetic diagnosis, but how receiving that specific molecular diagnosis can then enable precision medicine and positively impact healthcare management of these families. A few other examples where rare disease genome analysis has enabled precision medicine approaches at BC Children's Hospital. Though all of them are published, we do not have explicit consent from these families to delve deeper into their case reports beyond the publication, so I couldn't talk more about these, but in case y'all are interested, you can read up on them online. And that's it. Just a huge thank you to both the labs and the lab members that I work with. Everything that I've talked about requires massive team effort, and all these wonderful people really make it a whole lot easier. Yeah, just feel free to reach out to me if you have any questions or just would like to connect with me, and I can take any other questions that you may have right now. Thank you.
That was awesome. Thank you, Bhavi. Wow, you're doing some incredible work, and so are your colleagues. I don't come from a biological sciences background, but yeah, the way you presented that was very digestible, and it's really cool because in my personal life, I have friends who are kind of doing tests to see, I guess, the risk level of their children having some sort of disease or anything like that, and kind of understanding the background of what the work that goes into determining the risk level. That's really interesting, and yeah, awesome. Yeah, I guess, does anybody have any questions? Feel free to shoot them in the chat or just unmute yourself and chime in.
Bhavi, I've got a question regarding Canada's involvement in research around genomics and around. I was really surprised that a country like Estonia was putting more money into it than what Canada is.
Yes.
And Wales and Scotland. What is that about? Why would that have come about?
So the challenge with Canada, it's not that we're not putting in enough money towards this effort. The thing is, bioinformatics is also a very disruptive technology. So you need to have the right kind of support systems in place because you're going to get a lot of data. So you want to make sure that you can manage that data well, at the same time, protecting the patients and the families as well. The main issue with Canada was that a lot of these research on rare disease diagnostics that was happening was very project based, and these projects were focused on a particular type of neurological, like particular type of phenotype. So one project would focus only on neurological disorders, another would focus on only epilepsy specifically, even within neurological disorders. Some would focus only on kidney disorders. So even though there was a lot of progress happening on the research side, it was sort of disparate because it wasn't universal in terms of, like, it wasn't really looking at all rare disorders in general. And then it was broken down into research institutes. It was broken down into different research groups within these institutes. So we never got to the point where we were able to come to a common platform and share all the progress that we've made in terms of, addressing policies, developing frameworks that are needed. Because with rare disease genetics, it serves as a good primer to sort of develop the kind of infrastructure that you need, because you're working with smaller cases than what you would work with at a national level cohort. But you need that to come on a sort of a national research program at some point. And that happened very late in Canada. We are now slowly getting to a point where we are moving away from these disparate research projects, and we are focusing on more universal projects that are not just limited to one institute or one province. They are now Canada wide. So hopefully now we can address that, and then having built that infrastructure on the research side, we can hopefully then start translating that into the clinic.
Okay.
With Genome Canada's initiative, they're hoping that by 2025 we will have almost all rare disease genetic sequencing will be happening in clinics and will not be happening in the research side.
Okay.
And I can see progress is happening. In the last 2 years, initially I would get a lot of cases directly from the research side. That's not the case anymore, because now patients are getting approval from MSP in BC to do their exome sequencing. So it's directly going through the clinic and it's not coming to us. I've seen that the cases have slowly started trickling down to a smaller number, which is kind of bad because it's less work for me, but it's kind of good in a way that they're able to get the funding that they need from the government to pursue that.
Thank you.
Awesome. Thank you, Bhavi. So Nirali raised her hand earlier, and it looks like she has a question. Nirali, do you want to unmute yourself and ask?
Yeah, sure. Thank you, Heidi. And thank you so much, Dr. Modi, for this brilliant talk. So I have two questions. The first one is that like you have like diagnosed a rare disease in these two kids. So suppose like it was great that the mother came to you and you were able to screen. But if someone is having a rare disease, but they don't have any facility to go, how would like they would know about it? And second thing is that can we make this application in a way that is accessible to a larger population in a easy and less expensive way, so that more people are able to know that they have this disease, or they can use it in a better way, or we can know about, probably we can find more novel diseases like this. So do you like to comment on that, or probably we can like look forward to such kind of technology or something like that?
Sure. So I think both questions can be answered with the fact that that's what we are hoping to address with this national level initiative, right? That it becomes more accessible. Right now, it's happening through the research side. So yes, this particular family or the other families that we've helped answer questions on the research side is because they were able to come to BC Children's Hospital, which is very active in these kind of research programs. But if it was some other remote hospital somewhere, they probably would have to make the trip out to Vancouver to do that. So the fact that we want to move this technology, the thing that I say clinical implementation of genome analysis means bringing this into standard healthcare, meaning all doctors are trained, all doctors are known, they know when to be suspicious of a genetic disorder. They should know when to give them access to this kind of sequencing technology that they need. And all of that can only happen once you have a nationwide perspective, a nationwide plan in place, that access is available to every patient that needs help and that needs that access. So that will happen when it will move into the clinic. Once it's moved into the clinic, it's not going to cost anything to the families. It's going to be covered by a national, like the provincial health programs. BC has MSP. So, it's going to be covered like that. So it won't be at any cost to the family. It'll be completely covered by health insurance. And as far as the technology is concerned, that's also one of the other barriers why the progress in Canada has been slow, because we are kind of disparate in the way Canada's law is that even US for that matter, researchers that know how to do this, unless you're clinically certified, you cannot look at clinical data. So same is the case with me. Even though I know how to do this work, if a patient comes through the clinic, I cannot look at that patient's case because I'm not allowed to get access to clinical information. So if I want to really look at that patient's case, they have to be enrolled into a research program that I have access to, and only then I can do that. So there's this sort of a bridge that really needs to be addressed, because people who have the expertise and the knowledge are not able to do that because there's no mechanism to sort of clinically certify them to be able to do it. Or you need to train the existing force of clinicians to be able to do that. So that is another reason why this progress has been slow. But the technology is now being picked up at such a rapid pace that there are training programs in place. The Canadian College of Medical Geneticists has recently changed its syllabus to now include a lot of genome and analysis as well. So a lot of genetic counselors will have access to this kind of training. There are tools that are now available, like open source softwares. We do this in-house on Automodal, and it requires a lot of computing and coding. But for more simpler analysis, that there are commercially available softwares where it requires very minimal computing experience. So even a clinician can just put in the sequencing file and the tool will hopefully spit out a few variants for you. It's not perfect at this point, but it requires less expertise and less training in processing the data firsthand, if you know what I mean. So we're getting there. It's going, hopefully by 2025, we reach that goal of moving it into the clinic completely. It will be available and accessible to everyone.
Thank you so much, Dr. Modi.
Bhavi, yeah, it's unfortunate to hear that there's a little bit of that hurdle for people that are skilled in this kind of knowledge to get access to data that would really help the public. But hopefully that'll change soon with talks like this where you raise awareness about that issue. So we have two more questions. The first one's from Olga, and she says, "Thank you for the presentation. Could you comment on the autoimmune disorders and countries you think are ahead of Canada in terms of studying them?"
US and Australia are doing a really great job. There are local groups like Casanova's group in the US and Stuart Tangay's group in Australia are really kind of ahead in doing this, especially for autoimmune conditions and primary immunodeficiency disorders as well. Autoimmunity, the problem is a lot of immunologists still don't consider autoimmune to be a single genetic or monogenic disorder. So it really doesn't span in the rare disease space. It's kind of a complex disorder where there is interaction with the environment that also at play. So it's a lot more complicated and it requires a different analysis pipeline than rare disease genetic disorders, but sort of overlapping with autoimmunity are your primary immunodeficiency disorders. And these groups are certainly leading the way ahead of Canada. We're playing catch up, but slowly.
Great. Thank you, Bhavi. And then finally, I think Liana raised her hand. So Liana, maybe you want to unmute yourself and ask your question?
Thanks, Bhavi, for such a great talk. I think it was a really great walkthrough of all the resources it takes for patients to really get a diagnosis. My question is kind of post-diagnosis, thinking about therapeutic options for these patients. So, you know, shall a child go through this entire process, get their diagnosis? What are the options? I think in this particular case, it was kind of lucky that there was a small molecule inhibitor that happened to fit the solution. Can you speak a bit more about your thoughts on kind of potential drug development or drug solutions that could be developed for some of these rare diseases?
Yeah, I think it's a great question. Thank you. A big portion of this is what is known as the orphan drug development for rare disorders, because these patients, like all these drugs are considered to be orphan, and because they are so few in number, like you have 1 or 2 families that are affected at a time in one country. It's sadly, most pharmaceutical companies consider this to be a non-lucrative option in developing these drugs because it's just not that kind of volume generation. A lot of research is happening on biologic development or small molecule inhibitors, things that can really target the pathway essentially, specifically for that particular gene defect on the research side through the pharmacogenomic studies as well. But it still has been difficult in sort of moving that research and that progress into the clinic. Even for this particular drug, the side effects were low, so it was easy to get approval, and it was already approved for some other treatment, right? So we knew that the FDA approval and everything else has been taken care of, so it's easy to get I think there's a clinical word that I'm kind of not thinking, I cannot really think of right now, to get approval. It's on a case by case scenario, essentially, to just give the treatment to this family because the drug has been approved elsewhere. For a lot of the inhibitors and biologics that we discover or we think can help this any particular defect that comes up, unfortunately the cases that those drugs are not even approved for treatment for any other diseases as well, and that kind of really takes years at a time to get approval. Hopefully things will change with more funding being coming from the government for these orphan drugs, so that you're not really reliant on the private sector to develop them, because it's not a matter of just economics anymore. Obviously the private sector is very hesitant to pump in dollars and dollars of developing drugs for where there's just not enough volume. You're talking about sometimes there are cases where there are only maybe one or two in the entire world. So it really depends on the volume.
Great, thank you, Bhavi. So we have 10 minutes left, and it looks like we have one more question. Rachel, do you want to unmute yourself and ask your question?
Sure. Thanks so much for your talk. It was really interesting. I really liked what you were saying, talking about the kind of moving to a national initiative to make a big database, things like that. But in academia, especially with researchers that have been around for quite a while, they're a bit apprehensive, or I found they're apprehensive about the big data sets and gathering data from different sites and those kind of things. So they're apprehensive both about using other people's data as well as sharing their own. So I'm just curious if you can comment or if you run into that and how you kind of talk to them or reassure them.
Sure. I think that kind of has changed a lot in the last few years. And for me personally, especially because coming from both wet lab as well as dry lab backgrounds, a lot of the hesitance and the resistance that academics have usually comes from the wet lab perspective. For people that have been working in the dry lab or bioinformatics space, this has kind of become the common norm to rely on other people's data and use that data. Like you really cannot, at this point, the field of bioinformatics or even genome analysis is at a point where it's almost impossible to do anything without being reliant on other people's data. So for even one pipeline, I'm right now using over 40 different softwares and tools that were not developed by me. These include databases, these include softwares, everything. It's the number is more than 40 to 50 in one pipeline. So it's really impossible to do any research without having that kind of faith. So it's not really a question of trusting the database kind of structure or trusting that open source model of research anymore. It's really the question of what kind of access and what kind of infrastructure do you have to support that at this point.
Wonderful. I think that is it. Thank you everybody for asking those very, very in-depth questions. And of course, thank you, Bhavi, for the very insightful answers. That was a really great presentation. And
thank you so much. Thank you for having me.
Yeah, of course. You're obviously welcome.
Great questions.
Help out with SCWIST anytime and share your knowledge. I think before we wrap up, Christine has a couple of announcements.
SCWIST co-sponsors the Vancouver STEMs Book Club. And please go to Eventbrite and check when that is. It's on September 20. And we're raffling off a book. Can you see it? A book, Superior, which will be discussed on September 20. There you go. I prepared your names. I'm going to put it in a box. I'm going to shake the box and pull up a winner. Let's see. Yuyin won, and so I'll send you an email and give you instructions on how to claim your prize. Thanks, Heidi. I'm done.
Woo. Congrats, Yuyin, on winning a book. Always exciting. All right, so if you guys have any follow-up questions, feel free to connect with Bhavi directly, obviously, if it's a technical question, or if you have any questions about SCWIST, you can connect with me or Christine. Our contact information is on the SCWIST website, and you can also find us on LinkedIn. Thank you very much, and everybody enjoy the rest of your day.